I would like to now introduce Ibrahim Adam. Ibrahim developed a keen interest into becoming a biomedical scientist during his undergraduate study in Sudan. There he completed a Bachelor's of Sciences with honors at the University of Khartoum in Sudan. He then went on to get a Master's of Science in Genetics and Molecular Biology, where he developed an interest in immunology and then applied to the University of Alberta to get his PhD under the supervision of Dr. Laurie West. So thank you.
Thank you so much for the kind introduction. And thank you for the chance to present my work. Antibodies to non-self ABO carbohydrate antigen are produced spontaneously without sensitization. So if you are blood group A, for example, you would produce anti-B antibodies without any stimulation by blood or so on, and vice versa. If you're blood group O, you would produce anti-A and anti-B antibodies. And those ABO antibodies are natural antibodies, and they pose a very high risk of graft rejection in ABO-incompatible organ transplantation. And those ABO antibodies can also be induced by intentional stimulation, especially by ABO blood product. Although historically described as T-independent immune response antibodies, those ABO antibody, natural antibody has not been distinguished from the ones that induced by stimulation. And therefore, the role of T cell has not been conclusively determined. And the impact of sex on ABO antibodies has not been studied. And here we investigate the anti-antibody, natural versus induced, with the regard of sex and T cell requirement. So we ask three simple questions. Are anti-A natural antibody produced in the absence of T cell help? And can anti-A antibodies be induced or stimulated in the absence of T cells? And is there any sex difference in anti-A antibody production? To answer this question, we did series of experiments where, for example, we used wild-type mice or CD4 deficient mice, and we looked for anti-A natural antibodies by hemagglutination assay, and those mice were monitored by between 7 to 10 weeks old. And then we did some adoptive transfer where we added CD4 T cell into CD4 knockout mice. And then we looked for CD4 for anti-A antibody production after the adoptive transfer. And we looked for those mice between age of 5 to 10 weeks old, and then followed them up to 11 months old. In a third group, we use CD4 knockout mice and wild-type mice and CD4 knockout mice that has received CD4 T cells. And we sensitize those three groups with human type A erythrocyte, and we looked for stimulation of anti-A antibody production. First, we found that anti-A natural antibody production does not require T cell help because mice, wild-type mice in black, male or female, they produce anti-antibodies over time without any stimulation. But when we looked at CD4 knockout mice in blue, they produce even more natural anti-antibodies. So those mice don't have any T cell, suggesting that there's no T cell requirement for natural anti-antibody. And when we looked at female mice in red, CD4 knockout female mice, they produce very high natural amount of antibodies. So suggesting that in the absence of T cell, anti-A antibody production increase with striking sex difference. And then we looked for anti-A in CD4 knockout mice, male and female, over time. And, and we found that in the absence of T cell, anti-antibody production in female rise dramatically around puberty, usually around 4 to 6 weeks old, and then continue to produce more natural antibody as the mice get old. And the same for the male mice in blue, still they do not produce a similar amount as female mice in red. Then we looked at adoptive transfer by isolating CD4 T cell. And then we put those CD4 T cell from wild type mice into CD4 knockout mice. We use male T cell into male mice and female cells into female mice. And then we looked for successful transfer of those cells. And we found that addition of CD4 T cell into CD4 knockout mice suppress anti-A natural antibody production. The mice have higher natural antibodies. When we added CD4 T cell, those antibodies decreased, suggesting that those T cells suppress natural anti-A production. Then we looked for stimulation of anti-antibodies by stimulating the mice by human type A erythrocyte. That's a source of A antigen. And when we looked at female in wild type mice in black, we can induce anti-antibodies on those mice. The top graph for male mice and lower for female mice, they respond to stimulation by A antigen. But when we looked at CD4 knockout mice in male blue, we found that we cannot stimulate anti-antibodies without CD4 T cell because those mice, they don't have T cell and therefore they do not respond to stimulation by A antigen. They have a similar amount of natural antibodies and induced antibodies. Actually, there is no response to stimulation. And the same for female. Although female mice, they have a very high natural anti-antibodies, they do not produce more anti-antibodies in response to stimulation. You can see the red line, they have similar amount of antibodies, whether we stimulated or not stimulated them. But when we add a CD4 T cell in green for the male mice, now we can stimulate those mice. So those mice, they receive CD4 T cell and then stimulated by human type A antigen. And now they can respond to stimulation, suggesting that you need T cell in order to respond to human type A antigen. And the same for female mice, although in female mice, anti-antibodies are not stimulated beyond the high level of natural antibody. And therefore, we cannot see any response. We are not sure if those mice can really produce any more antibodies in response, or that's the maximum amount of antibodies produced. So in summary and conclusion, when we discriminate natural anti-A or ABO antibodies from that induced by stimulation, those findings we can see. First, production of anti-A natural antibodies does not require T cell. But in fact, T cell can suppress natural anti-antibodies because we saw that when we added T cells, they produce less amount of antibodies. And in the absence of T cell, anti-A natural antibodies occur in sex and age dependent, suggesting that there is a role of sex hormone. And now we're investigating which sex hormone may play a role in that natural antibody production. In contrast to natural anti-antibody, induction of anti-antibody requires CD4 T cell, so suggesting that it's T dependent. Thank you so much. I would like to acknowledge my co-workers and my supervisor and my colleagues in the lab, and I'm very happy to take any question.
Told you I couldn't wait for your presentation. Very nice. It's more a philosophical question, and maybe you addressed that a little bit in your introduction. So what is the role of natural antibodies, those natural antibodies, and why do you think there's that sex difference? Can you speculate on that?
As I will start with the first part. Natural antibodies, like they develop naturally. So anyone, like if you have, let's say, any blood group, you would have antibodies produced naturally. And those antibodies play an important role when it comes to blood transfusion or ABO-incompatible organ transplant, because if you have any anti-A or B, that would lead to hyperacute rejection in case of ABO-incompatible organ transplant, or blood hemolysis in case of blood transfusion. The role of sex, why is this sex different? We don't know. Maybe we suspect that sex hormone like estrogen might play an important role in why some like female mice have higher natural antibodies than male, because male and female, they have different hormones.
So maybe I didn't get it. So maybe I'm wrong. So please tell me. So do you have natural antibodies before transfusion or transplantation? Do you have them naturally?
Yes, so those natural antibodies develop usually by like about six, about 6 weeks old.
If we have them, there must be a reason, right? Because we're not born to be transfused or transplanted. So what's their role?
Yeah, so the common theory, they're saying that those natural antibodies induced by gut bacteria. So because when you start eating, then you will have a lot of bacteria in your guts, and they're saying those bacteria might induce those natural antibody. But it seemed to be like more complicated because we looked at gene-free mice, and still they have natural antibodies. So they must play some evolutionary role against maybe infection in babies and so on, at least in mice.
Quite interesting. Thank you.
Okay, sure.
That was a very nice talk. Can you, do you know anything about, say, differences post pregnancy? Like, are there differences in terms of anti-A and antibody titers in women who have had a baby? Like, is there, like, given, you know, differences in blood group, etc., of the fetus versus the mom?
For ABO, I'm not sure. Maybe Laura can answer that, but for rhesus, it's well known.
Yeah, so that has been a common assumption, I think, that, sorry, it's been commonly assumed that that women will have more anti-A and anti-B antibodies, depending on their own blood type, of course, that possibly could be stimulated by fetal exposure to non-expressed antigens. But in fact, now that we've developed a new tool to look at anti-A and anti-B antibodies more effectively and more reliably, and we're not finding that. So I think the whole literature needs to be re-examined a little bit, because it's been kind of sloppy literature over the decades. But as new tools have emerged to really assess these antibodies through Illuminex assay that we've developed now, you can really, we're starting to see much more clear information and precise and reproducible information. Using the hemagglutination assay, which is generally how people have done it for years, has so many substantial flaws in the methodology and the reproducibility that I think these things just haven't really been studied very well. And we're not finding that there is a higher elevation of antibodies after pregnancies, but we haven't had time yet to systematically look at individual pregnancies and so on.
I mean, you know, women also have a higher incidence of autoimmune disease, like autoimmune liver disease and autoimmune disease in general, right? So
yeah,
it's likely that estrogen has some impact in terms of, you know, that higher kind of immune state or heightened immune state, right?
Yeah.
Yeah. Okay. Thank you.
Yeah, those are really interesting things. So how do you think the CD4 T cells are inhibiting the antibody production?
So far, we don't know, but this data suggesting that at least interferon gamma produced by CD4 T cells suppress anti-A production in B cells. That's one of the theories. Okay. But we didn't test that, so I'm not very sure.
And do you know anything about the quality of antibodies, like the, you know, the wild type mice that have some, the knockout mice that have a lot? can you put that serum into a mouse with an A expressing graft, and does it reject a lot more? Or is it about the same, right? Like, is it more pathological, the antibody?
We did not do that exactly, but we sensitize wild type mice in order to produce a lot of anti-antibodies, and then we use that mice for hyperacute rejection. But we didn't look at natural antibodies, whether they would have the same effect. But that's very interesting question.
Well done. Yeah.
Okay, thank you so much.